So, yeah, so as Art pointed out, I'm going to give a presentation. I'm going to flip back and forth between some PowerPoint slides and between a web browser, maybe NMRbox. So I'm going to share my screen, and so I can bounce back and forth between some windows. Um, I'll talk about NAN at first, and then I'm going to talk about NMRbox. Uh, this is an acknowledgement slide. There's too many names to go through. Just you can see the vast number of people who've participated both on the NAN and the NMRbox project. And of course, um, NAN was funded by an NSF Mid-Scale Research Infrastructure 2 grant, and NMRbox is currently funded on a P41 NIH proposal grant. So here at UConn Health, we have sort of an umbrella of computational resources. We have the NAN project, which we're going to be talking about, and this is largely deals with instrument and the collecting of empirical data. NAN has existed for about 3 years now. NMRbox is the grant is in its 9th year, going into its 10th year. And NMRbox is a computational platform which just takes a lot of that empirical data and allows you to process it and analyze it and extract information. And then the Biomag Res Bank has been around since the 1980s, I believe. It was hosted by the University of Wisconsin, run by John Markley for years. When John Markley retired a few years ago, he thought because of the NMRbox project in particular and NAN starting up that UConn Health would be a good place to house the BMRB. So we relocated the BMRB, um, which is part of the Worldwide Protein Data Bank, to here in Connecticut. So Art touched on what the NAN deliverables were, and of course the two 1.1 gigahertz NMR instruments, the knowledge bases, and the knowledge bases are broken into two components: advanced knowledge bases and in the four subject matters that Art pointed out. We're also in the midst of just starting to build out what we're calling novice knowledge bases. This is information for novices to help them determine whether or not NAN or whether or not NMR would be a useful tool for them for the given research projects that they have. And then from there, you move into the advanced knowledge bases, which would basically be all the protocols and pulse programs and everything needed to collect the data. We also have what we call the NAN data transport system. So on the spectrometers, initially all the spectrometers at the first three sites at Georgia, Wisconsin, and UConn are basically harvesting data, and I'll talk more about that. As Art pointed out, we do want to start to see this system expand. We built out the feature set enough now where I think it would be great to see other facilities join in, which would allow a much larger data archive to be built. I'm also going to describe NAN Portal, which is basically a web resource, which has multiple components. It has a resource connector, which I'll show you. It has facility management and operations dashboards, which I'm not going to show you. I'll talk a little bit about it. Lab administration, again, I'm not going to show you. I'm going to stick with what the parts of NAN that a user would see. But I'll talk about it. The data browser we'll spend quite a bit of time on because it has, I think, a lot of great features. The advanced knowledge bases, and then UHF access request is something now that the instruments, the 1.1 gigahertz instruments are being built. We're in the process of creating access requests for users to submit applications to run data on the 1.1 gigahertz NMR instruments. So I'm going to start off with the NAN data transport system and not try to get into the weeds, but kind of give you a high level view of what NDTS is and how it operates. So the goal of NDTS is to automatically harvest data sets from the spectrometer. We decided to do this automatically rather than having users sort of save their data sets. we figured it would be better to just capture everything and sort through things rather than constantly be dealing with users forgetting to upload their data sets. So um, the other thing we need to do is we need to link the users and the PIs to those data sets. So primarily that's handled by NDTS. We're going to try to capture a lot of metadata automatically. Data sets by themselves aren't really useful unless we have a lot of metadata information attached to them. We'll obviously try to capture the user, we'll capture user entered metadata as well. There's certain types of metadata we cannot capture automatically. It would take user input for. And then, of course, the whole idea here, as Art pointed out, is to build an archive of well-annotated NMR data. There's a lot of federal grants that are collecting, that are being used to collect a lot of NMR data, and a lot of this data just gets trapped in lab computers, even people's own labs. Often PIs can't find data because, like a student kept it on a different computer or whatever. But really, all the data collected on federal research grants eventually should go into the public domain. So this is one of the things that we want to see is much better, much better data sharing. And NIH and NSF are both starting to crack down and require much better data sharing. So one of the things that we, as a facility joining that, is the users of that facility essentially will be satisfying NIH and NSF guidelines for data sharing. So the way NDTS works is there are essentially three main components. There is the workstation component, there's what we call a gateway computer, and then these are, say, servers at UConn. So on the spectrometer is a little software package that has actually two software packages. One of them sits and listens to VNMRJ or Topspin currently, and they listen for experiments to start and then stop. And when an experiment completes, then it basically is going to grab that data set, it's going to bundle it all together, and it's going to ship it to a gateway. A gateway is nothing more than a lightweight computer that sits somewhere in the NMR facility, or at least on the same network as the NMR facility. The gateway is then going to push that data to a server in UConn in our data center. That receiver is going to write that data to storage in what we call the landing zone. We have a data parser that's going to automatically parse that data, and the data parser is going to write entries in the database for that data set. So we're going to get an entry in the database for that data set. It's also going to write the data to primary storage, and it's also going to create a disaster recovery backup. And this disaster recovery backup is something that's written to a geodispersed file system across 4 data centers, and it's called a worm partition. You don't need to get into that, but it's basically the data can never be altered in any way by anybody, even system administrators or anybody. So we effectively, and then a third backup is actually coming online soon, which will be going to a tape archive. So we're going to have 3 copies of the data, one written in a way that can never be altered, and essentially the likelihood of data loss is very, very minimal. So one thing I will say is we'll protect the data better than what most facilities could do by themselves. Also on the data, the workstation is a little GUI, and I'll show some pictures of that in a minute. And this will allow users to interact with the with NDTS to control some of the settings. Some of the settings are actually the user controls from the web portal. So this is information when you log into the web portal, and you'll see this in a moment. You're going to be updating database records, and that not only is the data get pushed from the NMR facilities to the data center, but information that needs to get passed back will be sent to the receiver, and the gateway will also retrieve. So you see the gateway to NDTS receiver is a 2-way communication, and same way to the spectrometers. So the gateway will reach out and grab data from the receiver, and then the workstations will reach out and grab information from the gateway. And you'll see a little bit more about that in just a second. So I do want to say facility managers in our setup have a fair amount of control, and they have the ability to control like which workstation user on a spectrometer is linked to a NAN user. They get to determine the transport default settings for each user, whether a user can change the collection status, and whether a user can change which NAN username they have. So we built a system that's fairly flexible, but also allows the facility managers to control it, control the settings. And for instance, so maybe you're in an academic lab and your data should always be harvested. The facility manager can say, well, you're always going to collect your data. Or maybe the NMR facility collects proprietary data for a company, and that data should never be harvested. So they can also set it up so a workstation user's data would never be collected. And then they can allow users to change their settings as they like. So on the spectrometer, and I think you'll see this during the demo with John Gushka later, and I think John Grimes as well. When you're on the spectrometer, you see a little lightweight GUI that appears, and it gives you some information. It says that currently data will be transmitted to NAN, that the transport system is running and the top spin is running. It gives you some information about what probe is currently installed in the spectrometer, what the NAN username is, whether the harvesting is turned on and off, and if the user has permission, they could turn it off or they could turn harvesting back on. Then there's an experimental details button. So if the user presses the experimental detail button, then they get some additional, so this is sort of user-defined metadata they can add. And they can select what project the data set should belong to, what study, if there was a sample, what sample. And these project studies, samples, and usernames, this is the information that I spoke of that gets sort of pushed back from the NAN portal back into the spectrometers. And I'll talk a little bit more about project studies and samples in a bit. You get to say whether it's solution or solid, if there's the magic angles, and depending on which you choose here, you'll get some different metadata information that could be entered. If a data set was not harvested for some reason, if NDTS was toggled off and it should have been on, you can click a manual harvest button, you can select a data set, and then you can send it as well manually. As I said, one of the things that's very important is to not just collect the data, but to actually have a lot of metadata information that's linked with the data sets. So for any given experiment, we have several things that are collected automatically. We get all the files, and we don't just get the files in the experiment directory. We actually crawl around the file system and get additional files that Bruker and Topspin that are important, but which Bruker and Topspin don't necessarily include in the experiment directories themselves. And of course, these files include the parameters, the parameter files. We allow the user in NDTS to add additional parameters, tags, and notes. We derive and normalize parameters. That way, if you collect a different, so Bruker and Varian will define certain parameters differently, but we sort of have a normalized way of handling that. We have information about the facility, the instruments, and the probes, which automatically get linked to the data set. We know who the local user is and the NAN user. And then the user is able to select, as I said, information such as project study. The funding sources are associated with project and sample information. So I'll briefly talk about project studies and collections. So projects are created at the group level. So I'm just going to see. Yeah, you know, I'm actually going to skip and I'm going to come to permissions first, and I'll come back to project studies and collections. So, so NAN has actually a quite sophisticated permission model that lies beneath everything. But the basic premise is that data sets are owned by PIs. and data sets are associated with the NAN user. So now if the NAN user may be the PI, but in order to use NAN, we do have a requirement that every user either be a PI themselves or have their lab or have their account associated with their PI. And we do this so we can handle ownership properly. And we do vet every PI. So anybody who requests to be a PI, we actually go and confirm that manually. And then PIs get to control access to their data set. So the user can choose for people's own data sets, whether they can read or write, whether they can share it. But they can also control read the ability for people to read data sets or view data sets across the entire lab. So, and then we also know lots of PIs are very busy and won't bother with this. So we allow the PI to also designate a delegate who will then have basically full access and can act on the PI's behalf. We also allow the ability to do things like collaborative users. So in the model we have, everybody is going to be part of a lab. So, and then we have the idea of projects. So projects are created at the lab group or the PI level, and then everybody in the lab could be part of that group. And the PIs can add and invite the users to the project, and then you can add a funding source. So now the nice thing about this is if every data set you're collecting that's part of the project, if you link the project with that data set, then you have an organization of the data set, but you also have the funding for each of those data sets. You can also create studies. So studies are nested inside projects. And then inside studies are collections. So we have this nested sort of file hierarchy of project studies and collections. And studies and collections inside project can be created by any of the users. So the project itself is controlled by the PI or the PI delegate, but then the studies and collections within it can be done by any of the users who are invited to the project. We also allow personal collections to be created, and those could be done by any user. And as you saw from the NDTS GUI, you can select which project and study an experiment data set should belong to. And but after the fact, you're going to see you can group your data sets in projects and studies automatically. And for projects, we also have additional permissions. So if you want more fine-grained, if a PI wants more fine-grained control of who can do what to various data sets in different projects, they have control to do so. So now I was going to, instead of just telling you about the NAN portal, I thought it'd be better to just do a live demo and go through the resource connector, the data browser, samples, and the sample browser. So I'm going to switch over to I'm going to use an incognito tab here because I'm going to be using a test account. So in order to do this, we're just going to go to the nmrhub.org. And up on the right-hand side, there is a widget for logging in. And this would be true to get into any of our web services, not just NAN. I'm going to type the username and password in. Excuse me. And now I'm logged into NMR Hub, and you can get to the NMRbox website, the NAN website, BMRB, or NUSCOM, which is another project that we have. So we're just going to click NAN and visit site. And now I'll end up at the splash page for NAN. And you'll notice that we have two things happening here. We got a left menu button, which shows the ability to go to resource connector, operations dashboard, which I'll show in a moment, data browser, or the knowledge bases. And if you want, you can open and close that for screen real estate purposes. The splash page talks a lot about the same information that Art covered in his demo of what NMRbox really is. I'm going to click the resource connector, And from the resource connector, I'm going to go and do the facilities first. So right now, we have 3 facilities which are part of NAN, the University of Georgia, UConn Health, and NMR FAM. And you can see various information, what services are available at the facility. You can click a button that says more information about this facility, and you can get contact information about the staff. You can get links to the facility website and whatnot. So you also have filters on the side. Right now, with 3 facilities, there's not a whole lot of filtering needed, but the idea is this will expand in the future and allow users to be able to search through all the facilities that are part of NAN. You can go up to instruments. So if we come over to instruments, we see a tile for all the instruments that are part of NAN. And again, we have filters where if you wanted to filter based on whether it can do solid state, for instance, you can click that, or you can basically turn the filters off. And you can see all the instruments that are there. If we pick an instrument, say the 800 at CCRC, you can click on that and you can get additional information. You get a link to the UGA website where it has the weights, what the operating system is, current probe installed, and whatnot. So you can get additional information about the instruments. There's also an advanced search where somebody can do an advanced click on various pieces of information and the instruments will be sorted and only the ones that satisfy the filter would be shown. Again, currently with 3 facilities, this is a little bit much, but the hope is that in the future, we'd be able to get to a much larger number of instruments, and then these filters will be a little more meaningful. So now I want to go to the data browser. So for the data browser, what you see when you appear to the data browser is this. Again, we have this window here off to the left, and I can maybe open that up a little bit so we get a little more screen real estate. I get a little menu on the left, which we'll talk about in a bit. And then you see basically a table of data sets. In this case, they're being grouped into groups of 25, but you can show larger numbers, 50, up to 500 at a time. And then there's pagination windows to move through if you have a lot of data sets. And you'll see we get a data set name. These are just the directories of the data sets on the spectrometer themselves. Then we get information such as date, the instrument, whether it's public, pulse program, and others. And I'll talk about that in a moment. Initially, what you're going to see are all the data sets for which you have permission to observe. So you notice this first data set here is owned by the NAN user is actually John Glushka at UGA. And the reason I'm seeing it is because it's public. So I can see all public data sets. So I have permission to see all public data sets. So it's showing up on my browser. So you can see that there's a long list, there's a lot of columns we can see. And if you click the little wrench icon here, and you can get a pull down list of all the various metadata fields which are visible in the browser. And you can just toggle these on and off as you like. So I have some collected right now, but let's say you want a title, you can click on title. And now if I scroll, oh, well, here's the title. So for title is used very often on Bruker instruments for users to sort of write a note about the data set as they're collecting it, for instance, so we can extract that piece of information. Now, right now, I'm seeing a lot of data sets, one by John Glushka, a bunch by Yulia here, and I could page down and I could see some by Sasha. So but we have the ability to filter a lot of these as well. So we can, by default, the data sets are filtered by date. So the most recent ones are at the top. And I should say the reason why I see so many data sets is because this M-Test account that I've created is part of a group, the Jeff Hoke group. So in the Jeff Hoke permission settings, it says everybody in the lab can see everybody else's data. So I am seeing all the data sets that are associated with Jeff Hoke, and I'm also seeing any data sets that are public. But maybe I just want to see the data sets that are associated with M-test. So I can go to a quick filter and I can do my data. And now I see just the data sets that are owned by the NAN user mark test, for instance. And I could toggle that off again. And all of these columns have the ability to sort and filter. So let's say I wanted to filter and I only wanted to look at data that was contained NMR 600, for instance. I hit apply and I'd see only data sets from the NMR 600 instrument at UConn. And I can click this, I can do clear to filter, and it could disappear. Sometimes I might have, so I can also, same thing, contains, so in this case, the NAN user contains test. This is another way of filtering, so I only see data that's owned by myself. And this button up here allows me to clear all those filters. So sometimes you might have multiple filters on, and this button here is provided as a one-click way to just wipe all the filters away. You can also reorganize these things. So if you want the instrument over here, you drag and drop, and you can reorient them any way that you like. So, and that is saved as a user preference. So if you then log in on even another browser on another computer, the columns that you see in the order will be restored. Just looking at my notes to see various things that I'm covering here. Let's see. Yeah, you could do more sophisticated sorts and filters by adding different rules. So you can have multiple rules and you could do ands and ors. So you can imagine you get the whole Boolean logic. So you can do very sophisticated searches. For instance, um, I can look for pulse programs that have the word echo in it. And why I think there's there's a bunch here. So but let's say it's something I do all the time. I constantly are searching for those. So I can also create a new view and I can call it Echo Pulse Program and hit save. And now under saved views, it's there. So even if I clear all filters, I should be able to come and just do Echo. Oh, there is a bug. I just encountered, I just showed you one of our bugs that need to be addressed. In a couple of days, when we fix this bug, this would have just rerun that filter and it would only show me the pulse programs that have the word echo in them. All right, so in addition to all the filtering that could be done, we have a context menu. So if you click a data set, we can select it. And by holding down the control button, I can do multiple selects at once. I can also hold down the shift button and select a range of data sets, and then I can use control to deselect one. So you're allowed to select multiple data sets at once. And once you've done that, and I'm just going to switch to my data. So I'm only looking at data sets owned by myself. Once I do that, I can click a data set now, and with the right mouse button, I can get a whole context menu. So at the top, I can do an edited data set. So if there's any information, a lot of this information is locked because it's what was collected on the spectrometer. We don't allow it to be changed. But if there is information that can be changed, the user is able to edit it. I can also do a reassignment. So maybe this data set wasn't owned by me. Maybe it really should have been owned by, say, Harrison. I'm not going to hit save right here, but I could have selected Harrison and hit save. So we allow the users to reassign data sets to other users in their own lab. Facility managers have much more control to reassign data sets if necessary. We can download data sets. So maybe I want to download this to my local computer. I just click download and then I can hit save, and that data set was saved. I can open the folder. I can go to it, open it up, and there is the data set that was collected. So it's very straightforward from any computer to just download the data set or multiple data sets right to whichever computer you happen to be operating on. Another feature we have besides the downloading of data sets is NMRbox integration. So if I come and I do copy to NMRbox, I can do I set a directory, in this case UGA test, I pick a format and I hit save. And if I go to NMRbox, which we'll do in a little bit, that data set should actually already be there. As fast as it was able to download to my computer, it should have been pushed to my NMRbox home folder already. If you had processed data, if you processed the data on NMRbox, you can put data in a special directory called post-acquisition, and then you can retrieve that. So this is a way that you can actually take the processed data from NMRbox and bring it and basically suck it right into the data set. Those files then become a permanent record of the data set, and anybody else who ever downloads that data set in the future will also have those files included. You can also add supplemental data. So if you wanted to, that's not working. Oh, I'm sorry. It's showing me that there was already a piece of supplemental data that I did in testing associated with. There was a sample prep protocol that was a URL, but I can click add a new one, and you can select protocols, processing scripts, BMRB IDs, various pieces of information. And if you select one, so a sample prep protocol, you then can choose a URL, uploading a file, or a file in the data set. So let's say you had the prep for the sample was actually in the data set. You can click on that. It's grabbing all the files in the data set. Oop, no, it actually found an error. So you're seeing some of the ugliness here. We do have some bugs that need to be dealt with. But you get the idea. You can add a piece of supplemental data to a data set. So in addition to just getting the data from the spectrometer, users are able to add additional data information with those data sets. The data could be made public by default. All the data sets eventually will become public, but that doesn't happen for about 3 or 4 years after it is collected. And users have the ability to extend the dates for when data sets do become public. Samples we'll talk about in a moment. You can classify your data sets as to various categories, whether it's a calibration experiment, for instance, whatnot, and you can add tags and notes. We have the ability to publish data sets as well, and a data set that is published is actually copied. It is a new data set in our system. A persistent identifier called an ARC, which is a competitor to DOIs, is generated. And then, like, if you were publishing if you had a publication and you wanted to reference the data sets, you can provide a link to that ARC, and that ARC will always come back to the data browser to those data sets. So the last thing I wanted to point out here is redundancy. Sometimes we get data sets collected at the same time that have the exact same name. When that happens, we choose the most recent one to show, but we also indicate that there are 4 data sets, in this case, 4 data sets in that collection. And the star basically means that this is the one of the 4 that it chose to be the primary. There's actually two of them in this particular case, because I must have selected one in testing, that two of them to be shown. So you can see there's 4 data sets that are showing here, but I think we lost the filter. But the 2 data sets that were highlighted as non-redundant are shown. So now I think we'll move on to, I can show the project studies and collections. So over on the left, you can click all data sets. You have a test project. You can choose, you can break down into studies. In this case, these were just test ones for this here, but you do relaxation data sets, and then you can have collections underneath there, for instance, and you can click on any of these things, and you can see data sets that are inside any of these. And if you want a data set in any of these, you just literally drag it and drop it, and then it's part of that data set. I go there. So it's still always in all data sets, but it's now also associated with one of those collections. So project studies and collections are just a really nice way to be able to organize your data. So now I want to talk about samples. So I think you've seen in the data browser that we're getting all the data from the spectrometer. We're associating a lot of information with that we can search for, such as the instrument, the date, and pulse program and whatnot. But data without samples is kind of meaningless. So unfortunately, the only way to really get sample information is for users to enter it. So we've created a sample browser. So just much like the data browser, you can view all your samples in a table, and same way you can select which columns you want to observe and whatnot. And then you can create a sample. So you can create a single sample or multiple samples. In this case, this is a metabolomics workshop. So you would often be doing things in multiple samples. So I'll do an example of a multiple sample. So when you create a sample, you're going to get a form here, and you're going to have these headings across the top: the sample information, container, composition, properties, and additional information. So the PI is selected for you automatically. If you have multiple PIs, you can change the PI. The display name, you just have to give it a display name. You have to select whether it's solution or solid. A lot of the information here is not required. You can put things like dates, URLs, description tags, and whatnot. You can come over and you could choose information about the container. I'm actually going to come back to container in just a moment, but you can also do composition. So you can add components, and we have a wide variety of components that can be added. For instance, we have biomolecules, which are basically proteins, nucleic acid, carbohydrates. We have small molecules, and we have a wide range of choices to cover a lot of different samples. But I'm going to click a mixture and do biological material. So imagine this was like a urine sample for metabolomics. So I'm just going to call it urine. And then some of these values are not applicable in this particular case. And I can basically put a quantity. I can say each of the samples was 500 microliters. Isotopic labeling, not really relevant for a biological sample, so I can either choose unlabeled, or I could just leave something like that blank. If I come to container, I can basically say, in this case, they're going to be in tubes, and the tubes might be in a rack, and maybe this is where it's going to be variable. So I can select the tube label in the rack location. So by clicking this green toggle, I was able to highlight these two as blue. So these means that they're going to vary from sample to sample. I can add additional information if I want about the sample. I can go to sample properties, and I can measure, like, was the pH measured? Yes, it was, and it was pH 7. And you can fill out as little of this or as much as this as you would like. I'm going to not continue to fill it out for brevity. But when you're done, you hit close and continue, you hit proceed. I have to give it another name for the collection. In this particular case, I'm going to say we're going to auto-generate 5 samples. In this case, it made 5 samples with the name that I gave, UGA test, and a underscore 001 to 005. The 2 fields that I said were variable were the rack location and the tube label. So then this information would have to be filled out. You can download it as a CSV template, fill it out like an Excel, and then you can import it back in as well. I'm just gonna, for brevity, just gonna name these the same way. And then you hit create samples. And now under spawn sample collections, there was a collection of samples, and we just generated 5 samples that are all identical except for the tube label and the rack position in this case. So depending on how you're doing your study, you could, it may be different. I'm just doing this as an example here. So, and of course, we could have done this as a protein sample, an RNA sample. If you do a protein, then there's like an advanced protein window. So it's very, very verbose. The amount of information that you can add to the sample is very large. So I think a person could really detail every nuance about their sample in the system we have. But of course, almost everything other than a few fields are optional. So users can choose to do things it is to the extent that they want. So if you've created data sets, so if I come back to if I come back to my data sets and I come to my collections, I have a test collection. Well, let me show you that. So I'm just going to pick 5 data sets. So here's 5 data sets. And of course, these are not part of a metabolomic study. These are just 5 data sets that I'm using as a test. But imagine these 5 data sets were collected as part of a metabolomic study. I drag them into Metabolomics 2, and I have these 5 data sets sitting here. Now, I don't have any samples associated with them, but I can right mouse click. Now I can do multi-sample editing, and I can choose the 5 samples I just created. I can do next. I won't go into the details, but we have different ways of sorting the data sets in different ways, depending on how people are doing their studies. And in this case, there's 1 data set per sample, or each sample would have had 1 data set because I have 5 of each. I'll do 1-to-1. I hit next, and then I see the mapping of those data sets. And again, if this were done for real, they would be sorted in a more proper order than just 5 random data sets that I had chosen. But if they were misordered, like one could actually reorder them on the list, one could add new rows, one could switch the sample. So the system has a lot of flexibility, but the idea is that when you come to this mapping tool, hopefully they're sort of mapped for you just by choosing things appropriately. But if I hit finish, I hit yes, and now we can see that each of these samples are linked. So these are the data sets, the 5 data sets, and this little link here basically means that this sample is linked to it. And we can go back and forth. So UGA test 5, if I click this one here, I go to the sample browser and I can see that sample, or I can click this button and I can come back and see that data set. So we have very easily, you can migrate back and forth between samples and data sets. So that is largely what I had to show on the NAN sort of portal side of things. Hopefully you can appreciate sort of the power. Boy, I wish I had this when I was starting out, to be honest, because I have disks full of data sets scattered all over the place. And this is just a great way where all the data sets you ever collect are basically all sitting in one place, backed up securely and with lots of metadata information attached to them and lots of ability to sort and to organize and sort and filter through the data sets.
Hey,
So,
Mark,
yep,
I'm wondering if there are a lot of questions that have come in about that we'll get to in question answer. But if you're able to, could you do the link where you're gonna if you join NMR box now with a virtual machine and then or through your real VNC and then show how to get one of these data sets
yep,
into NMR box efficiently? That'd be really
yep.
useful.
Yep. So we'll do that. Yeah. The next thing I was going to do was to jump back over and show some things on NMR box. Um,
Perfect.
but so let me do that. Let me show a couple slides on NMR box, then I'll open NMR box and then we'll push some data back and forth.
And if we could do that in, let's say, 15 minutes or less, then we could have some question answer.
Sure. Okay.
Thank-
So I'll just do a very short intro. I have a very long talks that I can give on NMR box or NAN, but I'm trying to keep it short. So everybody should have an NMR box account, or hopefully everybody's at least working with support tickets to try to get their NMR box account created. Very briefly, NMRbox is just a platform that has a lot of different scientific software packages used by the NMR community in related fields. We are agnostic. We'll install any software that is requested by us. The software spans from spectral reconstruction, spectral visualization, you name it. You can read through the list here. Huge numbers of software packages. In addition to NMR, we also have software dealing with SACS, cryo-EM, molecular dynamics, and onward. We have a few commercial packages. We're very fortunate to be able to include MATLAB in all toolboxes with unlimited licenses. We're able to do this through the University of Connecticut's license to MATLAB, but we're able to offer that to the entire NMR box community. We have Mestranova, which is a commercial package for processing and analyzing generally small molecule data, and some other software packages. And then we try to pack as much stuff as we can in, such as editor's office tools and a lot of libraries. So people are trying to create their own programs. Hopefully, a lot of the libraries and packages that are there that are needed will be there automatically. When you go to the NMR box home folder, and I can kind of show this as a demo briefly in a moment as well, one of the pages you can click is the hardware page. And this will show all of the servers that are running. We currently have probably upwards of 50 to 60 servers running that users are connected into. Some of them you need to request access to. Anybody can have access to, but you just need to request. These are sort of our really biggest, most powerful servers. It tells you what the status is, whether it's online or offline. So sometimes you're trying to connect to an NMR box server and you can't connect. Well, go to the hardware page and make sure it's actually online. Sometimes we take them down for services. You can get an indication of which desktop you're logged into. So in this case, this example would show me logged into molybdenum because I have a little screen here. Tells you what release is currently running, how many days it's going to be up before it gets rebooted, and then information about um, resources and what those resource loads are. We run a rolling release model, so we run 6 concurrent versions of NMRbox. 1/6 of our servers are refreshed every 2 weeks, and so every server lives for 12 weeks until it's rebooted and rebuilt as a new version. And we're constantly stowing away what we call long-term support versions. So software won't always ever completely roll off the platform. Advantages of doing it this way is we don't need to do a full shutdown in order to do an update. Every 2 weeks, we can get new software updated and pushed onto the platform. And we can allows us to do much easier hardware maintaining. And then long calculations can be started at any time. But just make sure you start a long calculation on a server that's going to run long enough before it needs to be rebooted. The one disadvantage is there's heterogeneity across the software then. If you're trying to do something across multiple NMRbox servers at once, and you need the same version of the software, there's a little bit of complexity to that. But we do have some solutions to it as well. You can contact me separately about that. We have a software registry that lists all the software and information about those software packages. The Software Explorer is also inside NMRbox. So once you're in NMRbox, and I'll demo this very briefly, you can select the Software Explorer. This lists all the software packages, gives you information about the software packages, and importantly gives information about what commands you would use to run those software packages. When you're in the window, you'll see a little X up here or this X here. If you click either of those two X's, that will close the window. This is akin to turning on and off your screen. You can also log out. This actually logs you out of the system. So if you click either of these two X's, the screen disappears, but anything that was running continues to run. So you can go home, open it back up, and everything is where you left it. When you're done doing things on an NMRbox, you're best to log out. Makes things more stable if you log out, if you're really not doing anything anymore. One of the things you'll also want to do is go to Settings, Display, and you can change your screen resolution. That's very important. We had a lot of new NMRbox users here today, and we choose a screen resolution that's rather small, so everybody can, so we know you're going to be able to see the screen, but it's not going to be optimal. So you're going to want to go and change that screen resolution to something optimal for whatever screen you're looking at. We have way too many features in NMRbox to go over. I'll highlight a couple of them. One is called the Repository, which is a data lake of public databases, which is mounted directly in NMRbox. We have currently the BMRB, the PDB, NCBI Blast, Gizmo, AlphaFold. And so if you're doing a study and you want to do, like, you need BMRB depositions or PDB structures, they're actually already there. You don't need to go to the PDB and download them, though all the PDB structures are mounted in an NMRbox, and they're mounted in a way where if you're doing a study, you can basically use the lock version of the PDB. They won't change month to month on you. File locations. So the software that we install is always in user software. Everybody has a home folder. Most users' home folders are going to be in home, NMRbox, some number, and then a username. We're at over 6,000 users on the platform, so we needed to start dividing things up. That's why there's a number there. Note that every NMRbox has fast scratch. So sometimes your home folder is, the nice thing about your home folder is you log into any NMRbox server and all the files are the same. The downside about that is you get a little bit of performance hit. So if you're doing processing of data and you're seeing it act slow because you can't read and write files quick enough, you can do things on the scratch disk, which would be very fast. But note that it is truly scratch. So you need to copy your data back to your home folder when you're done. We have a public folder for sharing. You're able to create share groups to share data amongst your collaborators or your lab. There's tutorial data, and then the repository mount point, as I just mentioned. I'll go through that. For getting help, we have FAQs, we have documentation, there's support at NMRbox, which I know some of you have been using, trying to get your account set up. Inside NMRbox, there's a support button. On the website, there's support buttons, and we have a Slack workspace. So we have a lot of different places to find information and to give us support tickets. I do recommend, if you're going to file a support ticket, give as much information as you can. We get so many support tickets that just are I can't log into NMRbox, or NMRbox isn't working. But we have 60 servers and another 50 servers and that are running everything behind the scenes. We need a little bit more information to diagnose and help what the problem may be.